Hi everybody, my name is Igor, and welcome to a series called Public Research. In this series, we're going to take a look at some openly available articles online and do a first reading of them. Now, I've chosen an article of interest here for our first episode. It's really relevant to a lot of what's going on today, where bridges are being built between the languages of the world. We're all trying to listen to one another more than ever before and understand what it is that we're coming together towards. So, with that in mind, I've chosen an article here to talk about how when we even engage with a YouTube video like this, we're picking up on subtle cues that aren't necessarily just visual or auditory, but that our body language conveys as well. So here we go. Let's not fluff it up too much. Let's just do a first reading of this article together and pick out some parts that we understand about it. If you have any kind of questions or insights, I'd love to know in the comments, and let's just get started. So I have the article here from the National Library of Medicine. I found it on pubmed.ncbi.nlm.nih.gov /363282272. That's the link. I'm going to put it in the description. The title of the article is Speech Listening Entails Neuroencoding of Visible Articulatory Features by A. Pastore, A. Tomassini, I. Delis E. Dolfini, L. Fadiga, and A. D'Ausilio. Now we're going to read the abstract here and see how the article goes along. So, speech processing entails a complex interplay between bottom-up and top-down computations. The former is reflected in the neural entrainment to the quasi-rhythmic components of speech acoustics, while the latter is supposed to guide the selection of the most relevant input subspace. And I was curious here what neural entrainment specifically meant. So, neural entrainment is the synchronization of brainwave oscillations to external rhythmic stimuli such as speech, music, or flashing lights. It aligns neural activity with environmental rhythms, enhancing attention, learning, and sensory processing. This, described in and can also occur between individuals, enabling communication and social bonding. So here's a little bit of a tighter summary. It is the alignment of endogenous or internal neural activity, so the stuff happening in your brain, with exogenous or external rhythmic input, enabling phase and frequency locking. So all of the waves and signals bouncing in and around your brain are trying to coordinate and sync up with the waves and input that you're receiving from the outside world. So neuroentrainment is that lining up of brainwave oscillations with the outer world. Top-down signals are believed to originate mainly from motor regions, yet similar activities have been shown to tune attentional cycles also for simpler non-speech stimuli. Here we examined whether, during speech listening, the brain reconstructs articulatory patterns associated to speech production. We measured electroencephalographic data while participants listened to sentences during the production of which articulatory kinematics of lips, jaws, and tongue were also recorded via electromagnetic articulography. We captured the patterns of articulatory coordination through principal-component analysis and used partial information decomposition to identify whether the speech envelope and each of the kinematic components provided unique, synergistic, and/or redundant information regarding the EEG signals. Interestingly, tongue movements contain both unique as well as synergistic information with the envelope that are encoded in the listener's brain activity. This demonstrates that during speech listening, the brain retrieves highly specific and unique motor information that is never accessible through vision, thus leveraging audio motor maps that arise most likely from the acquisition of speech production during development. It's trying to tell us here that outside of what we're seeing and including what we're hearing, the tongue is actually having detectable movements that add to your understanding of what you're hearing. Now, I've pulled up using this link right here the full text of the article. It is hosted here on sciencedirect.com, published to the Neuroimage Journal, Volume 264, on December 1st, 2022. And then I further opened up here the PDF for us to read. The highlights of the article are that EEG activity entrains to speech articulatory synergies. Articulatory synergies contain unique information about speech. The brain reconstructs articulatory information that is not available to vision. And here articulatory being literally movements, so this is articulation, me moving my hand. And the speech auditory motor mapping may be built via speech production. And here's the full article. Now, I'll leave the schools here and the keywords for you to peruse on your own. I will include the links in the description. We're just going to go ahead and read the body of the article. And again, we've already read here the abstract. So 1. Introduction. Verbal interaction is an essential part of human behavior, and our brains are tuned to decode speech. Neural oscillations in the delta and theta range are believed to play a key role in shaping speech perception. Indeed, coupling of brain oscillatory activity to the quasi-rhythmic properties of speech, or speech neural entrainment, positively scales with speech intelligibility, and is tightly related to speech comprehension performance. Importantly, rhythmic neural electrical stimulation casually modulates speech comprehension performance, providing compelling evidence that the brain oscillatory activity is functionally relevant for speech processing. But see Zofel et al. 2018b for a discussion on alternative interpretations of the neural entrainment phenomenon. Brain entrainment to speech in the delta and theta band is suggested to increase comprehension via a facilitation of task-relevant information. The cocktail party effect is an example of this effect where selective attention translates into increased neural entrainment to the attended acoustic stream. Furthermore, if other speech-related cues are available, neuroactivity can also entrain to these signals. For instance, when acoustic intelligibility is compromised, oscillatory occipital activity couples to the periodicity of lip-face movements. It's saying here when the sound is difficult for you to fully capture, the waves, and that's what it means by oscillatory, and let's just confirm here. The occipital region located at the lower back of the skull, so it is the hind part of the brain. We can even go here. The occipital lobes. When acoustic intelligibility is compromised, oscillatory occipital, excuse me. activity couples to the periodicity of lip face movements. Your lip reading. Unsurprisingly, speech comprehension mostly benefits from visual cues in suboptimal listening conditions. Neural entrainment to speech thus reflects top-down influences, which are driven by prior knowledge and/or context to predict the temporal structure of the heard stimuli. One source of top-down modulation is located in the frontal lobe, as supported by the finding that oscillatory activity in the left inferior frontal cortex between 1 and 3 Hz and motor cortex between 4 and 8 Hz modulates the phase of low-frequency activity in auditory areas. And the hertz that it's talking about here are literally the electric signals and the waves that it mentioned earlier that are happening in your brain. These in the delta and theta range. This modulation may reflect a domain general mechanism extending beyond speech processing, with the motor system orchestrating sensory processing in time. Whether the motor system provides domain general temporal predictions or richer domain specific information about articulatory features is, however, still unclear. Indeed, top-down motor influences may exploit action circuits to implement an internal stimulation of movements. Let's specify here. A top-down approach is a method of breaking down a large system, project, or concept from the general big picture into specific, detailed, and manageable subcomponents, so from bigger to smaller. Or a hierarchical management structure where decisions are made by upper management. It is widely used in business, programming, and analysis. Top-down processing in the brain refers to cognitive goal-driven perception, where higher-level mental processes, expectations, knowledge, memories shape how sensory information is interpreted. It works by applying prior knowledge to fill in gaps in incoming data, acting as a shortcut for quick interpretation, often starting with a general holistic idea before focusing on specific details. Bottom-up brain processing, on the other hand, is data-driven, sensory-driven method where the brain builds perception from raw sensory input, so from little to big. Sights, sounds, feelings, before forming a full picture. It acts as a bottom-up flow where lower-level sensory information is sent to higher-level brain regions for interpretation, often described as automatic and quick. So it's saying here that based on the big picture, our brains are actually trying to form a prediction, so a simulation of, I keep clicking the references there, of what's about to happen. To investigate this relevant question, we designed an EEG experiment where participants listened to auditorily presented sentences. The sentences were obtained from a publicly available dataset in which acoustic data is synchronized with articulatory data recorded via electromagnetic articulography. Electromagnetic articulography, or EMA, uses miniaturized sensor coils placed on articulators, and we can see the points where they did so here, lips, jaws, and tongue, to measure accurate position data with a high sampling frequency, so very often, during speech production. Of key relevance to the current research is that the EMA provides the accurate description of speech articulators that is essential to uncover whether motor information contributes to the representation of speech in the listener's brain. To this end, we use the partial information decomposition method that is designed to separate unique, redundant or shared, or synergistic or complementary information provided by two source signals, here speech envelope and kinematic data, about a third target signal, here brain activity. We thus tested whether articulatory kinematics is encoded during listening and conveys information about speech that cannot be attained from the speech envelope alone, i.e., unique neural information about kinematics or synergistic neurorepresentation of speech envelope and kinematics, a better prediction of the neural response from both modalities simultaneously. Our hypothesis was that if speech-related neural entrainment entails also a domain-specific motor process, entrainment to speech kinematics will be observed. Here we can see the places where the sensors were picking up any kind of measurement. A total of 23 healthy, naive volunteers were recruited for this study and were paid 30, I think this is euros, let's see, 30 euros for their participation. All participants were native speakers of Italian, right-handed by self-report, and had a normal or corrected to normal vision. One participant was excluded because of technical problems during data acquisition. Analysis was performed on data from the remaining 22 participants, 13 females, ages 23.04 to plus or minus 3.44, which was the mean plus the standard deviation. Participants were informed about the experimental procedure and gave their written consent before participation. And because this is often so overlooked, let's go ahead and specify this too. Mean primarily refers to the average of a set of numbers, sum divided by count, or as an adjective, it describes someone who is unkind, cruel, or stingy. But we're not that mean here. We're looking at a different kind of mean. So mean is another word for average, which is just the total number of participants divided, yeah, divided by the total number being measured, so the age of the participants divided by the number of participants. The other two options they could have looked at here were the median, which is the exact middle value, and the mode is the value that appears most frequently in the dataset. The experiment was approved by the local ethical committee Comitato Etico Unico della Provincia di Ferrara. Approval number 170592. The kinematic principal components, the schematic of the positions of the electromagnetic sensors, and here we have the meaning of these acronyms. I'll let you all read this on your own. The stimuli were selected from the multi-speaking style articulatory corpus, MSPKA, which comprises simultaneous recordings of audio and articulatory lips, jaws, and tongue data of three mother tongue speakers pronouncing sentences in Italian. Audio was recorded at a sampling rate of 22.05 kHz. Articulators were tracked at a sampling frequency of 400 Hz by means of an electromagnetic articulography system. The EMA data provides a very accurate characterization of mouth kinematics and is commonly used in speech technology research. In the present study, we use data corresponding to X, Y, and Z positions of 7 sensor coils glued on the upper lip, upper jaw, lower jaw, tongue tip, tongue middle, and tongue back. For this study, we used 50 sentences, duration ranging from 6.2 to 9.4 seconds, pronounced by the same female speaker, referred to as 11s, within the dataset. The acoustic stimuli were manually checked and processed to remove any silent and/or noisy part at the beginning and at the end of the sentences. All acoustic stimuli were then normalized to the same average intensity of 71 decibels. Data corresponding to one sentence out of 50 were discarded from the analysis because the corresponding EMA data turned out to be corrupted. During the experiment, participants were provided only with the acoustic stimuli. The corresponding EMA data were only used for data analysis. Participants sat at about 80 centimeters distance in front of an LCD monitor, with their right hand resting on a button box. On each trial, participants were presented with a black fixation cross at the center of a uniformly gray screen. After a variable time ranging between 0.1 and 1.1 seconds, a randomly selected sentence was presented acoustically via two loudspeakers, placed at about 20 centimeters from both sides of the screen. The fixation cross was removed after a variable of time between 0.1 and 1.1 seconds from the end of the acoustic stimulus, and one word appeared at the center of the screen. The presented word rhymed 50% of the time with one of the words contained in the previously heard sentence. The word was selected to rhyme with any word in the sentence excluding the first, the last, and all monosyllabic ones. Participants had to indicate whether the word rhymed or not by pressing one of two buttons, located few centimeters apart, using always the same finger, the right index. The rhyming task was included to encourage participants to listen attentively to the whole sentences. To avoid possible biases in the participants' responses, we ensured that rhyming and non-rhyming words were matched for number of syllables and their frequency of use in the Italian language by means of an online software tool. Different words were presented for each repetition of the same sentence, amounting to 4 words for each sentence, 200 words in total. Every trial ended when participants provided their response in the rhyming task. Trials were automatically ended if no response was provided within 10 seconds. Participants were asked to reduce blinks as much as possible and maintain their eyes on the fixation cross for the whole duration of the sentence. The experiment consisted of 4 separate blocks of 50 trials, each 200 trials in total, with short in-between breaks. The whole experiment lasted about 2 hours, including the EEG cap mounting and preparation. Stimulus presentation and button press acquisition were controlled via MATLAB, and the Psych Toolbox 3 extensions. All relevant events in the trial start, stimulus onset, button press, were converted in a TTL by the ViewPix EEG system to accurately synchronize them with the EEG data. Here we see some of the measurements. EEG data were recorded continuously during the experiment with a 64-channel active electrode system. Electrooculagrams (EOGs) were recorded using 4 electrodes from the cap that were removed from their original scalp sites and placed bilaterally at the outer canthi and below and above the right eye to record horizontal and vertical eye movements respectively. Let's check quickly what outer canthi is. So it was the outside part of the eye. All electrodes were online referenced to the left mastoid. Presuming this is part of the... Okay. Just behind the ear it looks like. The impedance of the electrodes was kept below 10 kilo ohmegas. Let's see what this is. K omega. The impedance of the electrodes was kept below 10 k omegas. EEG signals were acquired at 1000 Hz. Analyses were performed within the MATLAB and Python computing environments using open source toolboxes and libraries such as Fieldtrip, MNE, and PID library, as well as custom-made code. Analyses, I think they meant analysis here. Analyses were performed only on trials in which participants gave correct responses in the rhyming task. 76.3 was the mean, with a standard deviation of 7.5%. In a range of 64.5 to 89.5. So the highest accuracy for an individual participant was 89.5, the lowest was 64.5. The average or the mean was 76.3, where they gave the correct response in the rhyming task with a standard deviation of 7.5%. The amplitude envelope of the acoustic speech signals was calculated by adapting a previously described method. As in the Chimera toolbox, we defined 6 frequency bands in the range of 80 to 8820 Hz that are equally spaced on the cochlear map. And this should be within the ear. So considering the cochlea of the ear, here's the cochlear map. We found something from near the top of the range here, all the way up to the apex, it sounds like. The speech signal was first filtered within those 6 frequency bands. Then we computed the absolute value of the Hilbert transform of each bandpass filtered signal. Finally, the speech envelope was obtained by summing up the result across all the frequency bands. The envelope was downsampled to 400 Hz to match the sampling frequency of the EMA data. To capture meaningful speech coordination patterns in the high-dimensional EMA data, i.e. 7 sensors by 3 dimensions, equals 21 times series of position data. We use a dimensionality reduction technique. We applied principal component analysis as implemented in the Fieldtrip toolbox. PCA outputs feature activations over time that explain part of the variance in the EMA measurements and are orthogonal to each other. Orthogonal describes objects, vectors, or functions that are perpendicular, intersecting at right angles, or are statistically independent and uncorrelated. Furthermore, PCA provides information about the relative contribution of each kinematic feature to the reconstruction of the EMA recordings in single trials. By visually inspecting the absolute values of the PC weights, it is thus possible to assess the physiological validity of the articulatory coordination pattern identified by each PC. Principal component. The continuous EEG data were bandpass filtered between 0.5 and 100 Hz, 2-pass Butterworth filter 4th order, and downsampled to 400 Hz to match the sampling frequency of the EMA data. Data were then re-referenced to the common average and time aligned to the acoustic stimulus onset from -1 second to the duration of the longest sentence plus 1 second. Data were visually inspected and noisy trials were removed. Independent component analysis was then applied to identify and remove artifacts related to eye movements and heartbeat. Noisy channels T8 for one subject were excluded from the ICA analysis and substituted by linear interpolation of neighboring channels. The total amount of trials retained for further analysis was 142, with a standard deviation of 21.7. So on average, out of the 200 trials given to each participant, this is the number that they were able to maintain, after removing things like artifacts and irrelevant components. To quantify neural coupling to speech production, we used mutual information, a measure of statistical dependence that captures any type of relationship, even nonlinear and non-monotonic, between two signals. Our aim here was to uncover the neural representations of the different kinematic components in the brain of the listener and quantify the contributions of these representations to the neural encoding of speech. To this end, we computed the MI between each, or mutual information, between each recorded EEG signal and the speech envelope and each one of the extracted principal components, the kinematic components. Before computing mutual information, we first removed 1.5 seconds after each sound onset for each trial to exclude stimulus-locked evoked potentials. This interval was selected to avoid any contamination from late event-related modulations and their slow return to the isoelectric. We then shifted the EEG signals forward in time by 0.2 seconds relative to the SE and PCs based on preliminary analyses, showing that such a delay maximized information between EEG and all the source signals. The time shifting is consistent with the assumption that stimulus encoding necessarily follows stimulus presentation. Furthermore, an extensive literature has consistently shown that speech-brain coupling, or entrainment, is maximal at comparable lags of about 0.2 seconds for brain activities following auditory visual speech by 0.2 seconds. More specifically, we cut the SE and PC signals from 1 plus 1.5 seconds relative to stimulus onset up to stimulus offset, variable length dependent on stimulus duration, and the EEG signals from 1.7 seconds relative to stimulus onset up to 2 seconds, 0.2 seconds after stimulus offset. Finally, all signals were padded, mirror padding, and then band pass filtered between 0.5 and 10 Hz. This relatively broad frequency range was set based on prior inspection of the power spectra of both acoustic and kinematic signals, as it encompasses virtually all of their spectral content. The choice of the high cut-off frequency is also consistent with evidence that coupling between brain activities and speech envelope is mostly confined to frequencies below the alpha range. Mutual information was then calculated using a recent implementation of the Gaussian copula mutual information method, which provides a lower bound of the actual MI and is robust to high-dimensional signals. We then focused on the contribution of each kinematic component and the interactions between speech and kinematic components to the neural encoding of speech. We thus employed partial information decomposition, a recent multivariate mathematical framework originally proposed in Williams and Beer to quantify and characterize representational interactions in the human brain. PID was performed using a recent modification of the original algorithms, which is based on common change in surprisal. PID decomposes the mutual information between a target variable and a multivariate set of predictor variables called sources. Indeed, if the sources are not statistically independent from the target, they will provide non-zero joint mutual information about the target, which, in other terms, indexes the degree of dependence. PID allows them to disentangle this information, parceling it out into information that is uniquely carried by each of the sources, information that is shared by the sources, and information that is accessible only when considering the two sources together. So they're taking a look at the breakdown between all of the measurements here and trying to figure out what is the information that is redundant. So if you think about it as a structural web from the source of the stimulus entering your brain all the way to the computation or the processing, the entrainment, the encoding that your brain does, and you separate each of those channels and each of those steps and each of those components, what is the information that is shared by the sources or redundant, and what is unique to each, and information that is accessible only when considering the two sources together, which is synergistic. Here, we consider the EEG measurements at each channel as the target signal. We run 4 different PID models, every time including as sources the speech envelope derived directly from the acoustic stimuli, and one of the 4 kinematic features, a different one for each of the 4 models. So they're also including here not only the auditory signal and the processing, but these kinematic features, so the articulatory components as well. Obtained from the EMA data through PCA, the decomposition yielded 4 outcome terms. EEG-SE, the unique information that the speech envelope carries about the EEG signal and cannot be attained from the kinematic PCI. EEG-PCI, the unique information that the kinematic PCI carries about the EEG signal and cannot be attained from the speech envelope. EEG-SE-PCI, or SYN, the information that the joint observation of the two predictors provides about the EEG signal that cannot be attained by observing each predictor separately. And the RED, the information about EEG that is shared by the two sources, thus reflecting a common neural representation of speech and kinematic component. If the interaction between SE and PCI is redundant, the information about the EEG that is carried by PCI can be obtained also from SE and vice versa. In other words, there will be no information loss if either the SE or the PCI is not available. In contrast, if the interaction is synergistic, neural information is encoded by the relationship between the SE and PCI. In other words, we would obtain a better estimate of the EEG signal by considering SE and PCI together rather than independently. They're really exploring the difference between just hearing me say, "I like that," hearing and seeing me say, "I like that" with a thumbs up, and whether there's mutual or, or, or information that is only available in one or the other. In other words, we would obtain a better estimate of the EEG signal by considering the speech envelope, which again is just that external stimulus, and PCI. Let's see if there's a short. I'm looking for the acronym for PCI here. It's the kinematic signals. So in other words, we would obtain a better estimate of the EEG signal, so what's happening in your brain, by considering the audio and the kinematics together rather than independently. Finally, unique information is carried by only one of the two predictors. For example, a significant UPCI, so the unique information that the kinematic PCI carries about the EEG signal that cannot be attained from the speech envelope. So if I just give you a thumbs up, but I don't say "I like that," a significant UPCI would suggest that the corresponding brain response can only be predicted by that specific kinematic signal and not by the speech envelope. In a first PID analysis, all the signals were pre-processed in the same way as described above for mutual information. Epoching relative time shifting of EEG data, including bandpass filtering between 0.5 and 10 Hz. After these pre-processing steps, signals for all trials were concatenated and copula normalized. So we're going to have to look this one up here. Concatenated means linked together in a chain or series, often referring to merging data strings, text, or files in computing. and copula normalized. Let's see what this one is. Copula normalization involves transforming multivariate data to have standard, uniform, marginal distributions, usually between 0 and 1, to model dependency structures separately from marginal distributions. To further increase the granularity of our description, we explored the temporal dimension by running the PID analysis across different lags from negative zero .2 sec to zero .4 seconds in 50 millisecond steps. We also performed a frequency-resolved PID analysis to evaluate whether the acoustic and kinematic features carry information at different spectral ranges. To this end, all signals were bandpass filtered by applying a sliding window along the frequency axis in the range between 0.5 and 10 Hz in steps of 0.5 Hz and with a frequency window length of 1 Hz. A separate PID was then applied for each bandpass filtered set of signals. The output values obtained both in the MI and PID analysis were statistically evaluated against surrogate data. The original relationship between the two signals for the MI, or between the target, the EEG activity, and the sources, the SE and the PCI, for the PID, was destroyed without affecting the statistical properties of each signal, including its autocorrelation structure. More specifically, the EEG activity at each electrode and for each trial was circularly shifted by a number of samples that was randomly selected between the value over 4 and the value minus the value over 4, where n represents the number of samples of the shortest trial. The time-shifted data were then submitted to the same processing steps as described above for the original data, before applying the MIPID algorithms as for the original analysis. MI was computed between the EEG and each SE or PC feature, and I with I equals 1 to 4. The same applies for the PID analysis, whereby 4 separate PIDs were run by including as sources the SE and one of the 4 kinematic PCs. This procedure was repeated 1,000 times, yielding a surrogate distribution for each participant and each information component. We then applied one-tailed cluster-based permutation statistics to test at the group level whether the original information values were larger than the mean of the surrogate distribution, the mean of the information values obtained for the circularly shifted data. In practice, all samples exceeding an a priori decided threshold. Here, p is less than 0.05 one-tailed for univariate statistical testing. Dependent samples t-test were selected and subsequently clustered on the basis of their contiguity along the spatial dimension. And if we want to break down what they're saying here, think about it like a math equation, where so far we've looked at and obtained certain values. We've obtained the sound value, the measurements of the kinematics, the articulations. So just me spinning my hands here without the sound envelope of my explanation. Um, those two dimensions, the brain oscillations that result from a brain hearing and seeing those dimensions, and they've taken all of those values and they've divided them into certain statistical tests and even shifted the calculations across each other to get these relevant numbers, looking for significant values. So if a certain measurement is consistently accessible no matter by where, from which electrode on the brain from the cap that the participant is wearing is found, versus a value that is only found even across 1,000 permutations on one electrode. It's pretty significant to know why is something like a movement or something like a sound affecting various brain regions at once or just one. So they're taking these numbers, they're making calculations out of them, and then they're using these statistical tests to generate more interesting measurements. So where were we? For univariate statistical testing were selected and subsequently clustered on the basis of their contiguity along the spatial dimension. Cluster level statistics was computed by taking the sum of t-values in each cluster. Let's just quickly, this is something that I hear often, so let's make sure. T-values are test statistics used in t-tests to measure the size of the difference between sample means relative to the variations in the data. So it's, again, looking at that, is something coming out on average or rarely, and what is the significance? A higher absolute t-value indicates stronger evidence against the null hypothesis, suggesting results are not due to chance. This sum was then used as test statistic and evaluated against the distribution of maximum cluster t-values obtained after permuting the original and circularly shifted data. At the level of participant-specific averages, 1,000 permutations. The p-value was finally calculated as the proportion of random permutations yielding a larger test statistic, compared with that computed for the non-permuted data. PID results were also statistically evaluated at the single subject level by computing, separately for each subject and each electrode, the probability that the original information values exceeded the 95th percentile of the distribution for the circularly shifted data. Resulting p-values were corrected for multiple comparisons across electrodes by controlling the false discovery rate. Neural entrainment to the speech envelope, as well as the lips motion, are well-documented phenomena. However, only a fraction of speech articulation is available to vision, while most speech-relevant information is, in principle, contained in hidden articulators, e.g. tongue movement. We here set out to investigate whether articulatory kinematics that is not visually available to the listener still conveys information about the produced speech that goes above and beyond that contained in the speech envelope. We recorded the EEG brain-wide activity while native language participants were listening to acoustic speech stimuli taken from the multi-speaking style articulatory corpus. This corpus contains simultaneous recordings of audio and kinematic data of the articulatory tract measured via electromagnetic articulography, while speakers were pronouncing Italian sentences. The dimensionality of the articulatory data was reduced by means of PCA, and the four PCs accounting for most of the variance were selected for further analysis to examine the relationship between the kinematics associated to speech production in the speaker and the listener's ongoing brain activity. Oh. The first four components derived from PCA explained most of the total variance of the kinematic data. Uh, Inspection of the components' weights shows that the first two components represented almost entirely movements of the tongue on the anterior, posterior, and vertical axes, respectively. Two of the movements that contribute significantly to articulation, such as that of the tongue towards and away from the lips or the palate, were thus automatically identified by PCA. Lower lip and jaw. Again, along the anterior, posterior, as well as vertical axes, as well as the tongue mainly contributed to PC3, which, despite explaining a smaller amount of variance compared to the tongue movements, appeared to capture another meaningful articulatory component reflecting most likely mouth opening and closing, lip protrusion, and lip-tongue coordination. Finally, a more composite mixture of articulators moving along multiple directions contribute to PC4 possibly reflecting complex tongue-lip movement synergies. The remaining components explained negligible amounts of variance, and their articulatory interpretation was less straightforward. These components were thus excluded from further analysis. Examples of the reconstructed time series for the four retained kinematic components, along with the corresponding speech envelope, are shown in Figure 2. The analysis of their spectral content reveals that all the kinematic components show spectral concentration over a low frequency range between 1 and 4 Hz. The speech envelope, instead, in line with previous evidence, is marked by relatively higher frequencies with a broad spectral peak between 4 and 8 Hz. So this was quite a long read here, but basically they're going more in depth into specifically the kinematic, and again, kinematic means uh, movement, um, and science works like this, where you could say sound causes a movement. It's moving the air particles. It can be, um, movements can make a sound, but our ears are not picking up very easily, at least, the movements that I'm making. So here it's breaking down the kinematic data rather than what they're calling the speech envelope, which is the acoustic data, and saying that the spectral analysis, which is the analysis of the frequencies generated by the measurements of the data that we're looking at. Uh, the analysis of their spectral content reveals that all the kinematic components show spectral concentration over a low frequency range between 1 and 4 Hz in the delta band. So the delta waves, um, they correspond to the delta waves in your brain, and the speech envelope instead, in line with previous evidence, is marked by relatively higher frequencies with a broad spectral peak between 4 and 8 Hz in the theta brain oscillations. Firstly, we evaluated whether the brain encodes the information contained in the speech envelope as well as in the hidden speech kinematics by computing the mutual information between the respective signal pairs. As expected, MI for the speech envelope is statistically significant than that obtained for surrogate data. Hmm. And maximal in two foci overlaying central electrodes and more laterally right temporoparietal electrodes. Such a topography is indeed very similar to what reported in previous works when quantifying neural speech entrainment with linear as well as nonlinear coupling. Metrics. Remarkably, we found consistent mutual information also for one of the analyzed kinematic features, which mainly relates to the antero-posterior movements of the tongue. Similar to what obtained for the speech envelope, largely increases over central electrodes and over right temporoparietal electrodes. The speech envelope and the tongue movements could, however, provide brain-relevant information that is either exclusive to each feature, fundamentally shared, or complementary between the two features. To disentangle among these different possibilities, we employed a computational approach known as partial information decomposition. The speech envelope carries unique information in the four different PID models. The topographic distribution of such activity is very similar across all PID models and closely resembles that already observed for the MI. Most remarkably, passive listening also entails neuroencoding of kinematic information that is not accounted for by the speech amplitude fluctuations, the SE. Specifically, the PC1 provides unique information that is consistently represented in the listener's brain. PC1 not only carries unique informational content but also interacts significantly with the acoustic information in a synergistic fashion. In other words, its combination with the SE leads to a net increase in information encoded in the listener's brain. We also observe significant synergistic information between the SE in both PC2 and PC3. Indeed, PID enables to uncover representational interactions in the listener's brain that cannot be directly observable in pairwise measures of dependence, suggesting that the listener's brain may integrate articulatory information with SE in a super additive way. Redundancy is then observed between SE and the first three PCs for the first and second significant cluster, for the first and second highlighting the informational overlap that exists between articulation and speech output. Excuse me if it's, you know, uh, getting, I'm pushing through here. We're certainly warmed up now, so I may be moving a little bit too quickly. I just want to thank you all for sticking with me in the video through up to this point. Um, so it's been about an hour. We're gonna read up to the end of the article, and I hope that you enjoy the series. If you have any feedback, I would really love to hear it. I would really love to um, know any articles that you would like read and looked at together like this in the comments. If you liked the video, thank you very much for liking it. Thank you very much for subscribing. Let's keep going. So, I think I did read it, but I will read it again. Overall, the same pattern of results is observed also when statistical evaluation is performed at the single subject level with a large proportion of participants showing significant USE and UPC1. All PID systems resulted in 14 subjects having at least one significant channel for the SE unique information, while PC1 unique information is significant in 8 participants. Very few subjects show significant unique information for the other kinematic components. Synergistic information between SE and PC1 is significant in 14 subjects, and fewer for the other components. A non-negligible number of participants also report significant redundant information between SE and the first three PCs, which is in agreement with corresponding group-level statistics. The results reported above indicate that the brain encodes a certain amount of information carried by articulatory kinematics that cannot be equivalently extracted from the speech envelope. To explore the temporal dynamics of information encoding in the brain, we repeated the PID analysis by systematically varying the EEG lag with respect to all sources, in line with results on mutual information. Maximal information for all the most relevant components is obtained at about 0.2 seconds lag. To further gain insight into whether information conveyed by the kinematic and acoustic signals is band-limited and possibly concentrated within distinct frequency ranges, we repeated the PID analysis in a frequency-resolved way. Figure 5 shows the outcome of PID analysis as a function of frequency for SE and PC1, for which we show significant unique information in the previous analyses. The information that is uniquely carried by the SE is clearly spectrally selective, with maximal values being observed at around 5 to 6 Hz, i.e. in the theta band. A different spectral fingerprint, however, marks the information that is uniquely contained in the kinematics, which is enhanced within a lower frequency range between 1 and 4 Hz, i.e. in the delta band. So, these measurements and what they're looking at here, let me just explain a quick thing. So, a function is when you have two measurements, and you can think of the measurements as the axes here. and various results along each of the measurements. The function is the line charting the measurements that are resulting from exploring the two sets of results. So the line that you see here is the resulting function between the measurements that they're checking. They're saying here that the sound envelope and the physical components show a functional outcome across various different measurements. A different spectral fingerprint, however, marks the information that is uniquely contained in the kinematics, which is enhanced within a lower frequency range between 1 and 4 Hz, i.e. in the delta band. Neural entrainment to speech originates from the integration of bottom-up processing and top-down projections from higher-order functional nodes in the brain to select and isolate particular signals of interest. Here we've reached the discussion part of the article, and we're going all the way back to that first word that we discussed, which is the synchronization of brainwave oscillations to external rhythmic stimuli such as speech, music, or flashing lights. And it's saying that when we synchronize our brainwaves to speech, it comes from your brain using bottom-up processing, so taking the little small signals, each of the sounds that you're hearing, each of the things that you're seeing, the the small parts of the whole, bottom-up processing, and top-down projections, so the big picture that's in your mind and the scheme, the established way that you understand the world, and using that top-down projection to simulate what that bottom-up information is going to result in. So neuroentrainment to speech originates from the integration of bottom-up processing and top-down projections from higher-order functional nodes in the brain to select and isolate particular signals of interest. So areas of your brain that are dedicated to those processes are picking apart the significant information from the bottom-up process to match to your top-down projection. Top-down projections based on context and a priori, so previous, knowledge, bias, bottom-up sensory sampling via predictive models operating at multiple levels. For instance, the neural computations run in the motor system may provide key top-down signals to isolate segmental or suprasegmental. So segmental means part. So suprasegmental, let's just take a look at this really quickly. Suprasegmental are prosodic features of speech that extend over more than one sound segment, such as stress, pitch, intonation, rhythm, and speech rate. So segmental must be just the singular vowels and consonants. The neural computations run in the motor system may provide key top-down signals to isolate segmental or suprasegmental cues. In fact, the sensory ambiguity characterizing the acoustic stream may partially be solved by unambiguous or less ambiguous endogenous signals, arising from the inherent rhythmicity in speech articulation. Yet, for such a claim to be tenable, one should be able to find traces of articulatory signals in brain activities of speech listening participants. Most importantly, neural activity should encode articulatory information in a way that is not trivially explained by the encoding of other tightly coupled speech acoustic features, i.e. mouth opening and speech envelope. Here, we show that vocal tract configurations are encoded in the EEG signal and that they contain information that goes above and beyond the one carried by the speech envelope. This result was obtained through the combination of a series of targeted novel approaches. First and foremost, first, First and foremost, participants listened to a set of acoustic speech stimuli for which synchronized articulatory data was available. EMA data is characterized by a spatial and temporal resolution that is not otherwise achievable by other technologies for speech kinematics analysis. Moreover, as it is customary in the field of motor control, we reduce the dimensionality of the data to derive a tractable number of signals explaining most of the variance. Yet this choice has also key theoretical implications since data reduction techniques may be used to extract physiologically meaningful data-driven patterns of coordination across articulators, i.e. vocal tract synergies, which have far greater functional relevance than the raw time-varying spatial positions of isolated articulators. Secondly, Secondly, we used a mathematical framework, partial information decomposition, that captures complex nonlinear relations among variables and decomposes these relations into atoms of information between the target and the sources. The PID can indeed extract unique synergistic or redundant information contained in the sources. As expected from the abundant literature on neural entrainment to speech, the PID analysis highlights that the speech envelope contains unique information encoded in the EEG data. The topographical distribution of this effect matches the one normally observed with other analytical methods with the involvement of central and right temporoparietal electrodes. At the same time, we found that the first kinematic principal component reflecting the antero-posterior movements of the tongue also carries unique information about the neural signals. The topography of this effect is confined to central and right temporoparietal electrodes. Interestingly, the synergistic interaction between the speech envelope and the first, second, and third kinematic components conveys additional information about the EEG data. PC2 represents vertical tongue movements while PC3 captures the coordination between mouth opening, closing, and tongue motion. In these three cases, the topography similarly shows a distribution covering both central and right temporoparietal electrodes. So here, we're really going over all of the data that we just read again in the discussion and making sure that we recall where the processing was happening for these specific representations of the data. The PID analyses also reported redundant information between the first three kinematic components and speech envelope, stemming from the obvious coupling existing between articulators, motion, and speech sound output. So it's reminding us that there's redundant information. So your brain is processing actually the same thing several times in several different places, while also not processing that redundant information uh, in other places. So some parts of your brain have redundant or the same as in another part of your brain, while some parts of your brain have unique information for each specific value. Importantly, we found a spectral dissociation that emerged in the frequency-resolved PID analyses. Lower frequencies from about 0.5 to 4 Hz appear to be relevant for kinematic-specific information, unique information provided by the PC1, in agreement with previous evidence that entrainment in the delta range originates from higher-level processes in frontal and motor cortices in particular. Instead, higher frequencies between 4 and 8 Hz contain unique information carried by the speech envelope, such as delta-theta dissociation is compatible with the idea that neural entrainment in the theta band is associated to the phonetic features that are critical for speech recognition. While the delta range entrainment is more closely related to the perceived acoustic rhythm of speech, overall our results offer new insight regarding the functional origin of the delta-theta dissociation observed in speech neural entrainment, especially regarding the contribution provided by domain-specific motor processes. In fact, the goal of our study was to investigate if speech listening does entail neural coupling to highly granular speech kinematic information that is not readily available to the participants. In our study, participants listened to auditory speech signals and were never explicitly nor implicitly exposed to the articulatory side of speech. Recent studies showed that brain signals encode missing information such as acoustic features when only silent lip reading is allowed. In this case, the tight audiovisual contingencies experienced during early childhood as well as throughout life offer a solid ground to explain these phenomena according to a Bayesian perspective and as the result of multimodal associative learning. Curious about what this is. I've never heard this word before. Bayesian inference is a statistical method that updates the probability of a hypothesis as more evidence becomes available. Very nice. Thomas Bayes. Let's see who this was. Thomas Bayes was an English statistician, philosopher, and Presbyterian minister who is known for formulating a specific case of the theorem that bears his name, Bayes' theorem. Bayes never published what would become his most famous accomplishment. His notes were edited and published posthumously by Richard Price. An American novelist and screenwriter. Hmm. Hmm. There it is. Christian Church is always laying down the groundwork for statistics and science. According to a Bayesian perspective and as the result of multimodal associative learning, in our case, kinematic data contains information that is neither available during the experiment nor ever visually accessible throughout life. It follows that neural coupling to unavailable information cannot be explained by the lifelong learning of audiovisual associations. Rather, we advance the proposal that speech production learning must play a key ontogenetic role in explaining our results. Neural coupling to tongue kinematics could still imply that at least part of the articulatory information is retrieved from speech acoustics. Recent investigations have looked, for example, at the temporal fine structure of speech. The recent study by Tang et al. Showed that temporal fine structures and speech envelopes are both coupled to brain signals in the same frequency band, 3 to 6 Hz, and suggested that TFS can be exploited by the brain to reconstruct relevant temporal information when speech is distorted. When you don't clearly hear what may have been said, temporal fine structure may be exploited by the brain. Let's not be hesitant to even look something like this up as we reach the end of the article. Temporal fine structure refers to the rapid high-frequency oscillations of a sound waveform within narrow frequency bands, conveying precise timing information through neural phase locking. Importantly though, SE and TFS appear to share the same informational content when it comes to temporal cues useful for speech segmentation. notwithstanding the obvious fact that speech acoustics contains more information than that conveyed by the envelope. The mapping between such an acoustic information and the articulatory space is tremendously complex. During speech production, different phono-articulatory tract configurations produce the same acoustic target depending on phonetic context. Known as the many-to-one mapping problem, it would force the brain to solve an ill-posed inverse problem when listening to speech known as acoustic to articulatory inversion. Hmm. The obvious fact that speech acoustics contains more information than that conveyed by the envelope, the mapping between such an acoustic information and the articulatory space is tremendously complex. I see. It's really curious to the scientists who wrote the article why our brain isn't just hearing sound. It's hearing all these other factors, processing information both redundantly and uniquely, including movement and lip reading, all sorts of factors. And the sentence that got me here is that there's a ill-posed inverse problem, um, known as the many-to-one mapping problem, where it must be difficult to measure and understand the rich and vast amount of data that your brain is actually creating when perceptually we're just hearing someone say a sentence. So mapping that, as we living creatures really love to do, is very complex. It may even start to become infinite or have various possible divisions. I mean, I always say when you start getting into decimals rather than whole figures, when you start going into negative numbers, you know, how far can I really zoom in? The page has a limit, but numbers really don't. Okay, let's keep going here and finish the article. As a consequence, how is it possible that information related to the coordination of an invisible articulator is retrieved and potentially partially reconstructed from speech acoustics? An answer to this conundrum could be that during development, the brain approximates a solution for this inverse problem, mapping intended acoustic targets back to vocal tract articulatory parameters to allow intelligible speech production. Indeed, infants explore how sounds are produced by experimenting the full range of their vocal tract configurations. Infants explore how sounds are produced by experimenting the full range of their vocal tract configurations. I guess it's saying here that because babies are new, they are always trying to, excuse me, it's saying that because babies are new, they're always trying to invent new speech. They're exploring, I have these muscles, I'm hearing these sounds, what can I build out of it? What can I make that's new? In support of this idea, automatic speech recognition models trained with both acoustic and articulatory data achieve better classification performance with far fewer examples than acoustic-only training regimes. These models recapitulate some key properties of speech production development and demonstrate that learning auditory motor mappings grants more compact and efficient representations of speech acoustics. As a consequence of learning audio-motor contingencies, speech auditory processing should in principle be tuned to capture those cues that allow triggering of endogenously guided reconstruction of missing articulatory signals. And again, endogenously here is going to mean from within. So, endogenously guided reconstruction of missing articulatory signals. As a consequence of learning audio-motor contingencies, so because you pick up schemes as you grow up of what you believe the world to be like, speech auditory processing should in principle be tuned to capture those cues that allow triggering of endogenously guided reconstruction of missing articulatory signals. To date, regardless of how fine-grained articulatory information is mapped onto the acoustic space, there was no evidence that the brain encodes articulatory configurations whose relevance is functionally dependent on the acquisition of speech production, and thus reflecting an intrinsically domain-specific process. However, a conclusive demonstration of this point will only come from future developmental research, i.e. by investigating the developmental trajectory of the auditory-based neural encoding of invisible articulators, or exploiting a multi-language approach, i.e. by studying similar phenomena. And here it's sort of musing that while we should be able to predict by what we hear the articulations that are related to the sounds, it says that to date, regardless of how fine-grained articulatory information is mapped onto the acoustic space, there was no evidence that the brain encodes articulatory configurations whose relevance is functionally dependent on the acquisition of speech production, and thus reflecting an intrinsically domain-specific process. So it's measured with empirical data. To date, we cannot say that actions correspond to speech. They provide uh, data and information that is related and that is used to build up an understanding of what was said and what was done, but it's not always specific that certain actions and certain speech envelopes will always coincide. A conclusive demonstration of this point will only come from future developmental research. Yet, the idea of a tight functional relationship between speech production and speech perception is not a new concept. Indeed, transcranial magnetic stimulation of the motor system produce specific and somatotopic effects on speech discrimination performance. A recent series of studies proposed a more detailed oscillation-based mechanism through which the motor system could have an impact on speech perception. Asaneo and Popel found synchronized brain activity between motor and auditory areas during a syllable listening task and successfully modeled the speech-motor cortex as an oscillator coupled to the auditory system. According to this model, neuronal oscillations observed in auditory and motor cortices indeed synchronize in a frequency range corresponding to the mean syllable rate across languages, about 4.5 Hz. Endogenous signals from the motor system would phase reset neuronal oscillations in the auditory cortex to align the most excitable states to the occurrence of expected events and/or changes in the speech stream, with benefits on perceptual and comprehension performance. Perception is an inherently noisy process, And in order to cope with speech-intrinsic, talker-specific, and speech-extrinsic, environment-specific noise, our brain needs to integrate and weigh multiple sources of information depending on their reliability. In this regard, when the acoustic signal is corrupted, the increased importance of visual cues translates into stronger entrainment to lip movements. Here, we provide a demonstration that neural speech processing can draw inferences based on highly granular endogenous domain-specific motor signals, whose relevance for perception necessarily derives from the acquisition of speech production capabilities. When the acoustic signal is corrupted, the increased importance of visual cues translates into stronger entrainment to lip movements. Perception is an inherently noisy process, and in order to cope with speech-intrinsic and speech-extrinsic noise, our brain needs to integrate and weigh multiple sources of information depending on their reliability. So, because just the audio or just the movements are not wholly reliable on their own, perception is this very complex process happening in our brains that tries to take all available factors into account to produce what you consider reality. Here we provide a demonstration that neural speech processing can draw inferences based on highly granular, endogenous, domain-specific motor signals, whose relevance for perception necessarily derives from the acquisition of speech production capabilities. Substantial progress in our understanding could come by providing a clearer picture on 1. The detailed acoustic encoding of the different articulatory synergies. 2. Whether those acoustic cues overlap fully or partly with articulatory information. And 3. To which extent information is then truly synthesized via endogenous processes. And I will read the last parts here. Thank you very much for reading the article with me. So, the funding for this work was supported by the BIAL Foundation Grant for Scientific Research 2020 to AT Ministero della Salute, Ricerca Finalizzata 2016 Giovanni Ricercatori, and Ministero della Salute Ricerca Finalizzata 2018 Giovanni Ricercatori to AD Ministero della Ricerca and the European Union H2020 to LF. The data and code that support the findings of this study are openly available in Mendeley data as DOI 10.17632/SVY 9M6987N.1. Declaration of competing authors declared no competing interest. A. Pastore was responsible for the methodology, software, formal analysis, investigation, data curation, and writing. The original draft was by A. Tomassini. Methodology, writing, original draft, writing, review and editing. Supervision. I. Delis was responsible for the methodology, writing, review and editing, supervision. E. Dolfini was responsible for the investigation. L. Fadiga was responsible for the conceptualization, writing, review and editing, funding, acquisition. A. D'Ausilio was responsible for the conceptualization, writing, review and editing, visualization, supervision, and project administration. The authors would like to thank Leonardo Badino for the many fruitful discussions that motivated this study prior to its completion. Supplementary materials associated with this article can be found in the online version at DOI 10.1016//j.neuroimage.2022.119724. And I'll leave the references to you all. Once again, I'm starting this series called Public Research. This is episode 1. We read the article Speech Listening Entails Neural Encoding of Invisible Articulatory Features. It is openly available here on Science Direct, was published in the scientific journal Neuroimage in 2022, so 4 years ago. And once again, the links are going to be in the description. I'm looking forward to your comments. See you all next time.